Hi everyone, my name is Miguel, joined by my 3 classmates today, and today we're diving into one of the most important tests in microbiology, the Kirby-Bauer test, also called the disc diffusion method. It might sound fancy, but it's actually pretty simple. It's how scientists test which antibiotics can stop bacteria from growing in hospitals. This test literally helps doctors save lives by choosing the right medicine for infections. Think of it like a battle between antibiotics and bacteria. This test shows us who wins. So, how do we actually do it? Let's go step by step. First, we need a pure culture. That just means one type of bacteria growing in a tube or plate. Then we dip a sterile cotton swab into that bacterial sample and spread it evenly across the surface of an agar plate, or better known as or commonly as a petri dish. We spread it evenly so the entire plate grows bacteria, like painting a smooth, even coat of bacteria all over the surface. That's called making a bacterial lawn. Next, we grab a small paper disc that are each soaked with a known concentration of different antibiotics. For example, you might know some commonly things known as penicillin or tetracycline. We gently place those discs on agar plates with sterile tweezers. Now that the plate is ready for incubation, we put it in a warm incubator at about 37 Celsius for 24 hours. That's roughly the temperature of a human body, which makes sense because how many of these bacteria come from human infections. When we check the plate the next day, something amazing happens. As you can see, around some of the discs, the bacteria didn't grow. You'll see a clear circular zone around them. Those are called zones of incubation. Each clear ring means that the antibiotic was able to stop the bacteria from growing in that area. The bigger the ring, the more effective that antibiotic might be. But we don't just guess. We actually measure the diameter of the zone with a ruler or a caliper in millimeters. Then we compare those numbers to a standardized chart made by the Clinical and Laboratory Standards Institute, other well known as the CLSI. That chart tells us whether the bacteria are 1. Susceptible, meaning the antibiotic works well; 2. Intermediate, it works somewhat; or lastly and third, resistant, it doesn't work at all. This system keeps the results fair and consistent across labs and around the world. Now my great classmates joining me for some questions.
Wait, so if 2 antibiotics have the same zones of inhibition, does that mean that they are equally effective?
Good question. Not necessarily. Each antibiotic diffuses differently, so we always interpret the results using the standardized CLSI chart, not just by size alone.
What happens if the bacteria show resistance to all the antibiotics we tested?
Then it's a serious problem. That strain might be a multi-drug resistant, meaning doctors have limited treatment options. That's why testing like this is so essential.
So the Kirby-Bauer test doesn't just work in the lab, it literally guides us to what antibiotics can help save lives in hospitals?
Exactly. It connects what we learn in biology labs directly to real-world healthcare. That's why understanding the Kirby-Bauer test is so important. Thank you guys for watching today's video, and I hope from the process and the steps to even to why it's so important, you learn something new. Have a great day and a good one.